So I'm going to tell you a bit of a story about why I care so much about following water around. We are at the AWI seminar series. So obviously I'm in biological sciences, but I'm hoping to bring you all along with me, if you're not a biologist, about why I care so much about rivers and streams. But first, I couldn't do this without a team behind me. So I do a lot of collaborative science. I'm really lucky to be here at UA, where I have a bunch of really great colleagues here that are interested in freshwater science. I've also been funded by the National Science Foundation, as well as a recent DOE award. And I also need to just give a big shout out to the CQ Network, which is a network of primarily early career researchers who all got together over Zoom because, as we all know, this was my desk circa 2020. I was super lonely. I was a postdoc at the time. Everything was really disconnected. I felt really kind of isolated and alone. I'm sure a lot of folks can kind of agree that that was sort of a really rough time for everyone. But thankfully what happened was Dr. Adam Wymore, who's at the University of New Hampshire, he reached out to a lot of us who think about rivers and streams and biogeochemistry to kind of build this network out. And we met virtually every week. We chatted about papers, we chatted about ideas. We ultimately have written about 4 papers. There's a book chapter that's in revision. It even resulted in a funded proposal. And I think that this really speaks to sort of my approach to science, that if you want to go fast, go alone. If you want to go far, go together. So if this is any lesson, especially to the grad students in the room, really lean on your community, and yeah, you never know where that will take you. But what we were really interested in as part of the CQ network was sort of revisiting these core ecosystem ideas that have been proposed like in the 1970s. So if I'm an ecosystem ecologist, I primarily focus on water, and a big person who has really inspired a lot of the work that I do is Dr. Gene Likens. So Dr. Gene Likens, he had this idea that, okay, maybe we can start to think about using stream water just like you would think about going to the doctor and getting your blood taken, right? They might give you some diagnostic information about what is happening in your body based on your pulse, your heart rate, your blood pressure. All of those things are these integrated measurements of your health, right? That maybe we could start to think about stream chemistry that ultimately ends up being like a pulse of that watershed as this diagnostic tool. So he had this idea. Fun fact, he actually went to the NIH with this idea. They promptly shut it down. We're like, absolutely not, because right, it's like diagnostic. He ultimately went to NSF and got that funded. And the big project that I want to just introduce today is this idea of the watershed ecosystem concept. So the watershed ecosystem concept is the idea that a watershed unit integrates all of the signals as water is carrying it downstream that you can measure at that outlet, right? So all the water is moving towards you and you're measuring that chemistry. They did this in a watershed in New Hampshire and at Hubbard Brook. And essentially what they did, in summary, right, is they found out that if you clear cut a watershed, like you tear down all of the forest, you remove all of the green stuff that's doing all that evapotranspiration, a lot of the stuff that's doing the nutrient removal, like trees, right? They need nitrogen and things like that. What you do is you clear cut that system and you shift the water balance. So you change that pulse of water that is moving downstream. Along with the water that it's changing in terms of the water balance, it's also shifting the chemistry of that resulting water. So what you see here is this concentration of nitrate specifically before and after the clear cut, right? It's pretty clear after you clear cut a system that nitrate increases. So this idea that you can use this watershed as this integrated metric of something that's happening on the landscape is sort of the core idea that we were really excited about. So what we started thinking about beyond this idea of the watershed ecosystem concept is sort of all of these different factors that are ultimately influencing, yes, you have this big watershed, but there are all these things that are happening upstream of you, right? You might have land use changes, you might have really fast microbial processes that are happening all at the same time. You also have geological processes that are happening really slowly on that watershed. You might have seasonal influences. So all of these different processes, these meteorological, biological, and geological factors that influence that chemistry that you might be able to measure as that heartbeat of that ecosystem are ultimately happening across really disparate timescales. So this is also something that was introduced by Likens and Borman. Borman was Likens' colleague. So this is not a new idea in ecology, but I think the difference being that now what we have is we have better ways of measuring water chemistry. So we don't have to just like send an undergrad out to like measure, take a sample. We can actually put in situ water quality sensors and capture what's happening at the watershed scale. So what I mean by this, right, is that if we think about just the meteorological conditions, right, you might have a storm. That's going to have a really strong effect at the diel or at the event scale. Whereas if you have shifts in that the water balance of your ecosystem, for example, as a result of climate change, that might not be evident until you're at this decadal timescale, right, in terms of the changes in the water chemistry. If we're thinking about biological influence, this is all relative influence, by the way. These fast-acting processes like metabolism, microbially mediated interactions, those are probably happening really fast or based on these seasonal components, whereas they might not be emerging in patterns that are evident at the decadal timescale. And then obviously geology matters, as much as I hate to admit it, but like things like groundwater levels are also really important, and same with weathering processes that are happening across these long timescales. So these are all things that are happening across these big timescales, and we have a ton of data about water chemistry. This is because this is such an integrative measurement of something, of ecosystem processes, of these signals of landscape change, there's tons of records of data. And so what's really cool is that these patterns are inherently nested within these timescales. So we could think about some, like the concentration and discharge relationships, so that pulse of that ecosystem at the event scale. So when a storm event happens, what happens to that water chemistry? We could start to think about seasonal components. How does that pattern shift? If you're thinking about those biotic changes at the seasonal scale, and then also long-term, right? Like these interannual comparative events of what is happening as a result of shifting water balance or precipitation regimes. So today I'm going to talk about two vignettes about one that we're going to talk about that I advertised, which is the Arctic aspect, where we're going to talk about event scale CQ processes and how this helps us understand landscape change. And then hopefully I'll have some time at the end to talk about some new data that a colleague, Dr. Carla Atkinson in the biology department, and I have been working on probably for too long before we've started writing the paper. So I'm going to start off by talking about some work that is funded by Arctic System Sciences up on the north slope of Alaska. This is going to be talking about the event scale. And then I'm also going to bring you down to the Itchway-Notchway Swamp, which will talk about sort of interannual concentration discharge relationships and how those emerge across space. So hopefully you all are along for the ride with me at this point. But I'm going to start off again by talking about this idea of concentration discharge relationships up on the north slope of Alaska. So you might be, okay, so a lot of people, I know a lot of people in the room, like Annie over here, is laughing because for the most part, a lot of people when they think Arctic, right, they think really cold and frozen and vast, and this is what it looks like for like a good portion of the year. Like that's true. But for about 3 to 4 months of the year, the Arctic does green up, right? So it does end up having this lush-looking landscape. This is primarily tundra, and water does start flowing in these headwater streams and rivers, meaning that it is actually functioning just like we would see a river, down here in the temperate system. The one thing that's different about the Arctic that I do want to introduce you to is that the Arctic is defined by what's underneath. So while this above layer greens up, what's really different about these systems is that they're underlain by continuous permafrost. So permafrost is technically defined as soil that has been frozen for at least 2 consecutive years. And when we talk about permafrost degradation, what we're actually talking about is this active layer, which is that brown cutout up there, getting thicker, essentially reaching layers of permafrost that by definition are no longer permafrost. So this is a big concern because this permafrost soil contains a massive amount of carbon, up to 1,900 petagrams of carbon. Does anybody know how many zeros are in a petagram? 15. Yeah, so I wish I had candy. I'd throw it at you. That's what I do in my class. Come to ecosystem ecology. It shouldn't be a surprise to anyone in the room that the Arctic is warming 2 to 4 times faster than the rest of the planet. So this becomes a really big problem when we're talking about this active layer thickening and accessing these deeper layers of carbon. And this big question, right, is what is going to happen to these big, vast stores of permafrost carbon as the Arctic changes and as the Arctic warms? Now, what happens when the Arctic warms is that a lot of other things start changing. So, for example, we get changing temperature regimes, right? So that's not good. We get a shift in hydrology in terms of when and where that water is actually falling on that ecosystem. Shifts in wildfires. These systems have been experiencing more lightning storms, and lightning likes to start tundra fires, which is not great. We have shifts in vegetation from like low-lying tussocky tundra to more shrubby systems, so the shifts in how carbon is stored, as well as changes in permafrost condition, both in terms of how deep that active layer gets during the season, as well as these collapses called permafrost, called thermokarst conditions. And this is also matched with a change in phenology. So not only that the Arctic is warming, but it's experiencing shifts in when that warming happens and when rain falls on these systems. So it's experiencing this vast number of symptoms of change, right? So we're going back to this diagnostic question. How can we diagnose what is happening to these landscapes? So our argument with this project is that we can start to use Arctic ecosystems, Arctic watersheds specifically, and the chemistry that is emerging from these outlets as this sensor or this diagnostic tool to understand what is happening to these changing landscapes. Essentially, asking the question of what is happening to this leaky carbon that's flushing off these systems that is integrated by the water that is moving towards you, or like at this outlet point on this slide. One thing that I do want to highlight is that these measurements that happen are often done in the Arctic either at this really small scale. So this is based on a meta-analysis that I did during COVID because I was bored. So at the plot level, this is what's dominating Arctic studies for the most part, especially when we're talking about nutrients at the plot level of vegetation. At this scale, what the Arctic data is showing is that these systems are relatively tightly cycled, that there's a lot of withholding of nitrogen because the plants it's such high demand, right? So plants really need nitrogen to grow, so they're holding onto a lot of that nitrogen. But when we contrast that to larger scale studies like that are happening at the really large watershed or big catchment, or even regional scale studies, what's happening is that a lot of nutrients are moving towards that outlet. In big river systems in the Arctic, they're starting to see increases in concentration and fluxes of important nutrients like carbon and nitrogen. So the question becomes, okay, how do we start to integrate the understanding across these two disparate scales that are telling us two different things about the fate of carbon in the Arctic? So our argument is that we can start to use this subcatchment or like intermediate scale watershed scale, which is understudied in the Arctic, to start to understand what's happening on the landscape. It's essentially the Goldilocks scale, right? So 3 bears. Okay, so the way that we do this is we go up to Toolik Field Station. If you haven't had the chance to go, I highly recommend. They have lots of snacks, they feed you very well, but it is on the north slope of Alaska, so you fly to Fairbanks, you drive 9 hours north, and there you are in the middle of the Brooks Range. So what we do when we go to Toolik is we use a tool called high frequency monitoring. Okay, has anybody in this room tried to chase a storm? Like sample during a storm? Okay, very few, but it's hard, right? It's not an easy task to do. First of all, you have to get to Toolik. You have to actually be there during the storm, which is a whole trek. And then secondly, like this can actually be relatively dangerous. Like high flow conditions in these systems can become a big thing, like you don't want to be out. So instead what we do is we use these high frequency in situ water quality sensors. These are absorbance based, and we've done this for the last couple of years. We have data since 2016. What they measure is things like in situ carbon, nitrate. We can also get TDN or total dissolved nitrogen, and we combine this with the discharge data. So we're doing the exact same thing that Likens and Borman did up at Hubbard Brook, where we're measuring the pulse of this watershed, but we're doing it every 15 minutes, which is awesome. We get such cool data. The things that we can start to learn from this information is these dominant influence on these biogeochemical fluxes. So what is actually coming off of this landscape and how is it changing across that field season? So we do this in a couple of different watersheds. I'm going to focus on two today, primarily the Kuparic River, which is sort of the quintessential tundra stream that you might have read about if you think about Arctic systems, as well as Oxarquoit Creek, which is just over the ridge and looks like this. So the main difference that I would consider between Kuparic and Oxarquoit Creek that you should know, like regardless of if you know these systems or not, is that Kuparic tends to not have that many lakes. It has a very few lake density, or low lake density, whereas that you can see in Oxarquoit Creek, there's lots of lakes in these headwaters as water is moving towards that outlet on that triangle. So these pictures are sort of what the sensor station looks like at Oxarquoit Creek. It kind of looks like a little robot. But again, like the main difference being that Ox has these sort of stream lake chains where the lakes are like beads on the chain. So this will become important later, but try to remember that Ox has lakes. So you might be asking, okay, Ariel, what are you doing with this high frequency data that you have from two different watersheds across all these different years? Like, what the heck? So what we do is we use event scale CQ from these DOC and nitrate regimes, pairing that with discharge in order to tell us something about the pulse of that watershed. So at the event scale, this is just like if you were to put your hand on your fingers on your wrist. It is ultimately a pulse that is resulting from an increase in flow. So what we do is we then put this on a log-log plot, and this is really nice. We can get some really cool data from it. And what I'm going to focus on is essentially the CQ model. So what we're interested in here is essentially just the direction of this CQ plot. So that is measured by this equation here, and the important part is that beta, or the exponent, which gives us the slope of that relationship. That slope can be categorized in a couple of different ways. So it could be positive, and that indicates this enrichment. So that means that as discharge is increasing, you're actually getting an increase in the concentration that is flushing from your system, right? So that means that you have this enrichment signal where you have a ton of material that you can move into your stream. You might see something that's more chemostatic, where there's no change in concentration with an increase in discharge. That just means that flow might not be a strong control on that constituent, or it's not based on the availability of that material for hydrologic transport. Or you could have a depletion signal where you're actually starting to see dilution, right? So you're seeing a decline in that concentration relative to an increase in discharge. So again, these are like the ecosystem's heartbeat. So what you're capturing really is, at the event scale, just this nice metric of, okay, what is the availability of that stuff that we're interested in measuring? So we do this. Obviously, this is data. So this is DOC data from a Kuparic 2017 storm, really nice event window. And essentially, we are able to categorize these storms into either any of these categories, right? Just based on that beta alone. So this storm, for example, is an enrichment signal in DOC. This is what we might expect from an Arctic tundra system, right? I told you there's a lot of carbon in these systems, and what is happening here is as soon as water falls on that watershed, or rain essentially, or precipitation, you see a flush into the river system, meaning that a storm event is driving a lot of carbon material transport into the river network. So one thing that I want to mention is, the active layer that I mentioned previously, it change throughout the season. So essentially what happens is that at the beginning of the season, right, you have a very thin active layer, everything's frozen. As you thaw, like the active layer gets thicker. So we initially thought, okay, like this dynamic system is going to reflect this seasonal pattern. That CQ patterns, that heartbeat of that ecosystem, will be evident in these seasonal changes. And I'm just going to bury the lead here and say that no, there was no seasonal pattern at all with these CQ responses, which was fairly surprising to us. I'm not going to talk about that result too much today, but if you are interested, we published that in Limnology and Oceanography. But what I do want to talk about is the fact that we were also sort of surprised by this seasonal pattern, but then we started thinking more about, okay, well, how does the landscape context sort of set the stage for these CQ responses? So we started thinking about, okay, how could we start to think about this system that is quintessential tundra, right? Like this Kuparic landscape that's very, very organic rich, as well as this lake-based system where we have water that is moving not just in the stream network, but also kind of mixing in these chains of lakes. So the lakes being these alternative like reactors or amplifiers of those systems. So we then put this data, so this is the nitrate CQ slope on this axis, a DOC slope on these axes. Hopefully these categories make sense, but anything in like those quadrants basically depicts whether or not carbon or nitrogen is depleting or enriching. So this is essentially our data of all of our storm events from about 2017 through 2021. And one thing that you'll notice, right, is that there's a very clear signal from the Kuparic, all of these blue points, this tundra-based stream, that there's a carbon enrichment that happens concurrently with a nitrogen depletion. So this is actually really exciting, right? Because this means that when you have a storm event, what you're doing is you're exploding the amount of carbon and you're depleting the amount of nitrogen. So you're changing the stoichiometric ratio between C to N at the event scale. Now, Oxyrhynchus, the orange one here, tends to be slightly more buffered than the Kuparic. Again, this is the lake-based, the lake-dominated watershed. We think that this has to do with that water moving in and out of those stream-lake chains, where water tends to, mix within those highly reactive lake zones, and then, amplify and buffer, and then ultimately things just end up sort of being skewed towards more buffered. So one thing that I also think is really exciting about these data is that despite which watershed you are actually in, what this suggests is that a storm event essentially could exacerbate nitrogen limitation in these systems. I know I talk a lot about carbon, but nitrogen is a highly in-demand nutrient in these ecosystems. And so when we think about these storm events occurring, what we're doing is we're not only shunting a bunch of carbon, which it was previously on the landscape, right? We're putting a bunch of carbon in a watershed network where it is exposed to things like degradation, where it could end up as CO2 into the atmosphere. But we are also potentially moving a lot of nitrogen from an N-limited landscape and moving it into the watershed network where it is then shunted downstream. So this is important, right? So this idea of lakes acting as this amplifier or filter is something that I'm really excited about exploring. So this is just what I mean by that Oxyrhynchus kind of is this important indicator of what's happening on the landscape. So this is Ox. Here's a closer view of all of the lakes further up in the network. We have a lot of data, not just from high-frequency sensors, but also from grab samples from pretty much every single one of these lakes, including the inlets and the outlets. So, oh gosh, well, colors went away on that. These are just a couple of these lakes, basically increasing in lake volume. And one thing, I know it's not always useful to put a whole bunch of bar plots up for you, but one thing I want you to notice is that there are some scenarios where DOC is produced, meaning that there's more DOC in the outlet than there is in the inlet, and there's some lakes where DOC is removed and nitrogen is produced, meaning that the opposite trend is occurring. So spatially, because water is alternately moving between the stream channel and these lakes along this watershed network, I think this is a lot of the explanation as to why we see these sort of more buffered CQ patterns, meaning that what we are actually measuring downstream at that watershed outlet might not necessarily be reflecting all the complexities that are further upstream. And so might be this type of scenario where we can't actually use the CQ patterns necessarily to understand the landscape drivers of these emergent properties. So I'm going to skip over some things just for the interest of time, because I do want to make sure I get through some stuff. But one thing that I also think is really exciting in thinking about like Arctic systems, and something that I haven't mentioned yet, is the fact that storm events in the Arctic are especially expected to become more intense as climate change progresses, right? So there's this question as to not only how do these streams flush that carbon from the landscape into these river networks and are therefore exposed to export downstream, but how does the intensity of that storm sort of drive that carbon export? So one thing that I've been working with a postdoc, Chao Song, he's now faculty on this question, and we essentially took the CQ slopes and we divided it by the discharge quantile. Essentially, like, is it lower, like a less intense storm, or is it like a super extreme storm? So these are the same type of data. So CQ slope, when it's above 0, it just means an enrichment. If it's below, it means depletion. So essentially for the Kuparic, across years, something that you'll notice is that in general, with the exception of 2019, because 2019 was kind of a weird year, we start to see an increase or a shift in the CQ response based on the quantile. So the larger the quantile, the more extreme that flow event was. We see the opposite trend for nitrate, where nitrate tends to be more diluted the stronger the event is, right? So this matches the pattern that we were seeing before, but helps us understand that the intensity of that precipitation is actually what's driving the CQ response, meaning that the stronger the event is, the more carbon you're moving, the more nitrogen you're depleting, right? That makes sense. It's very intuitive. And ox, always the problem child, tends to be the opposite trend, where we don't see any clear influence of flow, with the exception, again, of 2019, because 2019 was decided that it wanted to cooperate with our hypotheses, I guess. But yeah. So the point being that the more water you have, so the more intense these storms become as the Arctic shifts towards this intensified seasonality of the hydrologic cycle, the more mobilization, the stronger that pulse will be of carbon mobilization and nitrogen depletion. So one thing that I think is really critical for us all to understand, so you might leave this talk saying, like, I don't understand a thing that she just said, like, it's okay, I apologize for that, but I do want you to leave this talk knowing something about these systems, right? So in Arctic systems, this timing of this initial flow, this freshet period, is going to get earlier. And the intensification of these late season storms will happen further and further back into that flow season. So what does that actually mean for the carbon and nutrient availability as this phenology shifts? I think that's like a major question that I think everyone, even people who don't think about the Arctic all the time, should start to think about. So I think this is a really important issue. And I also think that the thing that I think about is not just in terms of, okay, like, yeah, carbon is going to move downstream and maybe it'll be metabolized to CO2, but it's not just the ultimate fate of that carbon, right? It's also what is the stream environment experiencing as that storm event occurs. So something that I think that I want to underscore here is that carbon to nitrogen ratios are super important for ecosystem processes. So things like nutrient uptake, how the algal communities respond. So the fact that we're blowing up the C to N ratio, so we have a lot of carbon availability, but we're declining that nitrogen concentration, means that we are sort of underpinning the stoichiometric balance in these systems. So I think these questions are really exciting, and I hope that you also think they're interesting as well. So, okay, so I just took you to the Arctic, so let's warm up a little bit. I want to also just introduce you all to the Ichwe-Nachwe Swamp, which is this is a data set that we've been working on for a little while now, and is part of sort of the Stream Climbs project that is led by Dan Allen at Penn State. So this is, again, not going to think about event scale, but we're going to think about interannual CQ patterns of this unique system. So again, this is a warmer system, and there's a lot more water for a lot longer in the period of record. So these are my colleagues. This is Chelsea Smith and Dr. Atkinson for scale. Obviously, these are really, really wet systems. And I think they're really fascinating. And the thing that I appreciate what's the most about being here at UA, and something that I actually didn't know, I'm from Wisconsin, so I'm not from the South. I don't think about biodiversity hotspots in the South, or I didn't before I moved here. But I think it's something that's worth appreciating, that the southeastern United States is this biodiversity hotspot. And so we are number one, maybe not at football this year, but we are in terms of aquatic biodiversity, right? So I think that's a really important thing to highlight. And this is really supported by a lot of the water quality that we have in our stream and river ecosystems, right? Not just in Alabama, but also throughout the Southeast. So despite being a water-rich region, right, I think we all think about, it just rained a ton yesterday. Like, I think we all think about this as a very water-rich region. There is an extremely high water demand throughout the Southeast. And it's only increasing as a result of shifting priorities in terms of agriculture, society, like what cities need water availability, as well as other things like ecosystems, right? Like streams need water to function as streams, usually. So this is a map of counties in Georgia and just the irrigated acres in 2010. What you'll notice is that this part of Georgia is heavily irrigated, right? So what that does is it shifts the water demand towards putting more water, not necessarily in the stream or the swamp, but on the fields. So what does that actually mean? So this is a really great paper from Steve Galladay, where essentially this is caused by center pivot agriculture, right? So as soon as we put in center pivots, so the ability to take water from either groundwater, which ultimately ends up in the stream, et cetera, what you do is you shift the water balance, right? So you decrease the capacity or the discharge capacity of that river. So a lot of this is driven by like peanut agriculture and other ag, but this is a pervasive issue primarily in this region of Georgia. So this is a region that's it's going to be exposed to a significant amount of water demand in the future, both for agriculture, and it's also part of the tri-state water war, right? So if we think about this region, this watershed, this larger watershed, is controlled by 3 different states, right? So Alabama, Georgia, as well as Florida. Each of these states has different priorities in terms of how they use their water. So up in Atlanta, they want to be able to take showers in the summer because it's hot and they get sweaty. In Alabama, we want water that makes it to the ACF River Basin. And in Florida, they also want fresh water, water quality to be good so that they can have oysters, right? So there's multiple interacting factors as to why water demand is going to be so important in this region, but I think it's worth mentioning. So we're gonna focus in on the Southern Flint River Basin for the remainder of today's talk. So hopefully I can get through this. Are you ready? Okay. Okay, so this is the Itchway-Notchway watershed, where I have been really lucky to work through as part of the Stream Climbs project. Again, I am a Midwesterner at heart. I am not a swamp person. Like, this is very, very new to me. So I was able to work with Dr. Atkinson, where we essentially, got from the Jones Center hydrochemistry, so water chemistry, as well as discharge, since 1993. So it's almost as old as I am in terms of the data record, and we got it from 6 different sites. So water is moving this way towards site 6. So all of these sites, we have combined water chemistry and discharge, so basically monthly. So this is a massive data set that's super exciting, especially as a new faculty member who's like eager to look at like these long-term data, right? In addition to this, we also have this record of that water balance that I was talking about before. So this is the Palmer Drought Severity Index. When it is above 1 in this green, what it is showing is that that's a wet year relative to, so precipitation relative to ET is relatively high. If it's in this orange, this indicates drought conditions where ET is essentially outpacing precipitation. So this record helps us understand the climactic drivers that might potentially influence those concentration discharge responses within this watershed. So I just want to note that this is a weird place, like really weird to me. There's no defined channel. I got there and I was like, what is happening? This is really weird. Like I'm used to streams that are like nice, they have banks. This does not. Like this is really low relief region. So I just want to note that this is a relatively unusual place to work, but it drains the Chickasawhatchee Swamp Complex. So it's this really wonderful habitat for biodiversity. It's also this really amazing place to work that is, home to a lot of like that biodiversity that I tried to introduce at the beginning of this section. But this watershed, because of that interaction between humans and climate, is experiencing extreme wet and dry cycles. So this is one of the gauges. Chelsea took this picture, who's sitting in the audience right here, where it's flowing, it's looking good. And then this was, when was this, Chelsea?
Probably like
July of 2022. What you can see in this picture is that this has sort of become disconnected. It's stopped flowing, at least detectable flow, right? So this system has almost like not completely dried, but they do start to like experience this really, really low flows, and a lot of that is due to this center pivot ag. So there's a lot of pumping for irrigation. About 40% of this watershed is ag. So this is kind of an interesting question, right? Of like, how does this climate and water demand influence these emerging concentration discharge relationships that we can do spatially? So one thing that I just want to mention, so this was published in Ecological Applications, is we combined some of this data and ultimately found that there's distinct water chemistry that emerges as a result of these flow conditions. So when PDSI is low or negative, meaning that there are dry conditions, that means that we have a very, very low C to N flux. Whereas when we increase the PDSI, we have a higher C to N flux, which is interesting. And we can do this across all of these different sites. I know this is a lot of data, so please ignore a lot of the panels, but the point being that the more water you have, the higher C to N and higher C to P flux that you have from this watershed, meaning that dry and wet conditions do matter for the experienced chemistry. If you're an organism like living in the swamp, those flow conditions really do matter. So we started thinking about, okay, well, how does that change the CQ patterns, right? Like that heartbeat of that system, does that emerge at the scale of the watershed? So these are CQ slopes that will be divided by ET versus, or Palmer's Drought Severity Index by color. Oh my gosh, I don't know why the color disappeared on these, but essentially what we see is that there's no difference between the Palmer's Drought Severity Index across all of the different sites. Meaning that across all the different sites, across all the flow conditions, we see a ubiquity of nitrogen dilution, which is not too surprising. We also see a relatively common pattern of phosphorus enrichment that does not depend on flow conditions, and carbon enrichment, which also does not depend on flow conditions. So this was a little bit surprising in terms of what we were thinking about in terms of the availability of that carbon. But then what we started thinking about is, okay, what does the actual CQ relationship capture, right? So what we're talking about, this interannual concentration discharge relationships, we're essentially regressing this cloud of data. So we are trying to represent this entire watershed essentially using a single number. What happens when you have a lot of variability, or you might have some reason that concentration discharge relationship shifts at a given discharge? So what we can do instead of using a single number is use breakpoint analyses to assess where might we break that CQ response. What that does is it gives us an indication of at a certain flow, that watershed, that pulse has sort of shifted. So this would be like, okay, you're walking, you're running, and at a certain point, like your heartbeat's really high, and you've kind of maxed out like your heartbeat, right? So this is essentially the same thing, but just thinking in terms of the concentration availability of what you have to export in your watershed. So this helps us try to understand more of the nuanced dynamics of what's happening that's emerging from that watershed. So just to give you a little sneak peek, again, this is data that we're still kind of like thinking about and like trying to figure out the story behind this. But this is data from Site 1. So this will be discharge on the X and then DOC on the Y from Site 1. So this is essentially this relationship between PDSI, or with PDSI noted in the color. The thing that you'll notice is that there's no distinct driver of CQ based on the PDSI, but what there is that there's a significant breakpoint in that CQ response. So what's happening is that once you have a little bit of water on your watershed, it starts to flush, right? It's a strong enrichment signal, at least in this case at Site 1. And then it reaches a threshold where potentially it's maybe it's like reaching a certain point on the riparian zone. Like again, these are low elevation systems. And then it becomes a little bit more chemostatic, where it's a little bit less likely to mobilize a lot of that carbon, because you're kind of at capacity for that source. We do this obviously for all of the sites. Again, this is a lot, so I just want you to think about site 1 through 3. So at site 1, potentially this driver is a result of the reconnection of that floodplain. At a certain discharge, you start to elevate your water level, and then you start to flood it. And then that's kind of, you're reaching your capacity for that carbon sourcing. Site 2, the swamp being swamping, and so there's very little floodplain potentially as you move to this site, or there's just so much variability that there's no significant relationship between that concentration and discharge. And then site 3, very similar. I think this has more to do with like that reconnection of the floodplain, and then at a certain discharge, you're just over capacity in terms of flushing a lot of that carbon from this watershed. We can do the same thing for nitrogen. So we think about this site 1 again. This is the only site where wet versus dry conditions, so that PDSI matter. I'm not sure why that is yet. Again, I'm still exploring a lot of these data, or we still are. Site 2, again, I think the swamp is swamping, and it's just doing its thing. I don't know. I don't think that there's like a really strong relationship with Q, probably because Q is hard to measure right there. And then site 3, again, that floodplain is reconnected. So all of a sudden, like when you have lower Qs or less intense discharge, you're diluting your system, and then you reach a capacity where you can't dilute it anymore. It becomes a little bit more buffered, a little bit more chemostatic. You're just more limited in your ability to deplete your system. So all that I want to say is that there's no clear response for CNRP when we do this linear response, right, across these years. But what's really exciting is that we can start to dig deeper into that CQ response, think about the breakpoints and how that reflects sort of this landscape aquatic connectivity and when the floodplain actually is reconnected hydrologically in this low relief region. And something that I didn't get to that I would love to talk to someone about, or anyone about if you're interested, is that the antecedent conditions do seem to matter though. So while we don't see a clear pattern of CQ responses based on the PDSI or the drought severity, what we do see is that depending on the year prior, that helps predict that immediate year of what's happening. So if you have a really strong drought, that has a really strong, the year prior, that has a really strong control in that CQ response. So you might be asking yourself, all right, why do I care about these swamps, right? So this region, again, is really important, not just for like ecology. So obviously I care about this because this is super biodiverse. It's a really cool region. But it's also this precedent for water rights in the Southeast, right? So this is a really important scenario where the water quality of these ecosystems is important ecologically. It's also important for human land use implications and how we use our water responsibly, especially when we think about ag increases and climate shifts. So just to end, I know that I'm running up on time. I want to make sure there's time for questions. So the things that I told you today, I talked about two really disparate systems, right? Like Arctic systems are super different than anything here in the Southeast, including sort of the Itchue-Notchway swamp. But I think that this idea of watersheds responding to climate change, it becomes this really big, complicated system, or like this big, complicated problem to understand how systems are going to shift as a result of a lot of competing drivers. And like I was mentioning at the beginning of my talk, these drivers are competing across timescales and across space. So it's this really interesting, exciting problem, but I still think that watersheds are these really unique systems that can at least tell us something about how systems are going to respond because they're not closed, right? They're impacted by what we do to them and what is happening on the landscape unit. So all that to be said is that I think that these CQ responses, or at least water chemistry itself, allows us to be creative in how we do these analyses and the types of signals that we're looking for that are relevant across timescales. If you are interested in this paper about CQ responses, this just got published in Wire's Water. And yeah, I would happily send you a PDF. I think it might be behind a paywall, unfortunately. Yeah, with that, I will happily take any questions. And thank you for your time and attention. I really appreciate it.